Hello, everyone, and welcome to today's live broadcast about colony picking in a fully automated DNA assembly workflow, presented by Dr. Hilla Takota, Dr. Ivan Yuen, and Dr. Valentin Zulkor, all from the Edinburgh Genome Foundry. We are excited to bring you this educational web seminar hosted by LabRoots and sponsored by Molecular Devices. Molecular Devices is one of the world's leading providers of instruments, software, and reagents for life science research, pharmaceutical, and biotherapeutic development. Their broad product portfolio includes automated microbial colony pickers, which will feature in today's presentation. The team at Edinburgh Genome Foundry, EGF, have worked closely with Molecular Devices Automated Workflow Engineering Solutions, or AWS group, who can tailor Molecular Devices products according to individual customer needs. You can find out more about the AWS group by visiting the Molecular Devices website. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Dr. Takota, Dr. Yuen, and Dr. Zulkor. Dr. Hilla Takota led the design, installation, and establishment of the robotic platform at EGF. She currently leads a team of 7 specialized staff to develop fully automated DNA assembly for research and commercial purposes. Dr. Takota's background is in molecular and cell biology and has always included state-of-the-art technologies for cell and high-throughput biology. Dr. Ivan Yuen has a PhD in molecular and cell biology and has been involved in the setup of all instrumentation in the EGF. He is a talented biologist with expertise in script writing, and he has a very good feel for the delicate equipment on the platform. Dr. Valentin Zulkor has been developing software for computer-assisted design and manufacturing at the EGF since 2015 after a PhD in systems biology from the Université Joseph Fourier and engineering degrees in applied mathematics from École Centrale Paris and TU Vienna. He is also a former professional GIF maker. Please join me in welcoming Dr. Takota. I will now turn the presentation over to her.
Thank you very much for this opportunity to present the Edinburgh Genome Foundry. We are a newly established facility for synthetic biology at the University of Edinburgh in Scotland. DNA assembly is one of the key processes to enable synthetic biology, and this can be easily automated. We would like to give you an overview of how this is done here at the EGF. We will put special emphasis on our customized Qpix Select HT colony picker and spreader from Molecular Devices that you can see here in the foreground. A crucial step in DNA assembly is the amplification of DNA in bacteria or yeast. The Qpix can plate out these cells on agar dishes and the next day pick individual colonies for growth. The Qpix is integrated into our robotic platform, and we will describe the process in more detail. Before, let me just briefly present synthetic biology and why this field needs platforms like the Edinburgh Genome Foundry. The Foundry's primary focus is synthetic biology. Synthetic biology is about using genetic parts to reprogram organisms to achieve useful functions. The field has expanded a lot in the past decade, and I will give one example of what it can do. On the left here, you see the plant Artemisia annua, also known as sweet wormwood. For a long time, artemisinin has been isolated from this plant and been used in traditional Chinese medicine. Artemisinin has also been found to have antimalarial properties. However, It is not very practical and cost-effective to isolate enough artemisinin to treat all malaria patients with a plant isolate. The research group of J. Kiesling was able to reprogram yeast to produce the antimalarial drug artemisinin back in 2014. This required transforming the whole genetic pathway to produce this valuable antimalarial drug precursor in yeast. This demonstrates the enormous capacity of synthetic biology in the production of pharmaceutical agents, but it is also applicable to the production of biofuels, cosmetics, biosensors, and much more. Developing new synthetic biology products is a trial and error process that typically takes years. It is generally broken down in 4 phases: design, build, test, and learn. In the design phase, genetic circuits are designed based on requirements and prior knowledge. This produces the parts list and sequences of the DNA to be assembled. In the build phase, the circuits are assembled from parts, using one of several available DNA assembly techniques. The assembled constructs are then quality controlled and examined for their function in the organism. This is also known as the test phase. The new experimental data then will be analyzed by bioinformaticians or systems biologists to refine models and estimations, and therefore expanding a knowledge base and allowing for better designs in the next iteration. A constant trend in the last decade has been to try and automate each step of the synthetic biology process. Automation allows for lower error rates, higher throughputs, higher precision, cheaper production, and much more. The build and test phases can be automated using robotic laboratory equipment and computer-assisted manufacturing software. The learn and design phases can be semi-automated using machine learning or automatic design programs. The automation of an experimental cycle was first performed by Ross King and his lab in Manchester in 2004. Their setup, the robot scientist, was able to execute an experimentation loop and generate hypotheses from the data. This set the path for more advanced robotic setups like the Edinburgh Genome Foundry. At the Edinburgh Genome Foundry, we perform all steps of the cycle, but we focus in particular on the build and test phases. Our robotic setup is designed to accommodate many different DNA assembly methods, and we also have a fleet of robots for quality control and phenotypical assays. The learn and design part of the process are generally led by our customers and their specific research demands. As a university facility, we are here to provide the automated build and test as a service. The Edinburgh Genome Foundry is part of a network of foundries that were funded by the Research Council UK as part of their Synthetic Biology for Growth program to specifically accelerate synthetic biology projects in the UK. While having similar technologies, each foundry has different biological areas of expertise. For instance, the Earlham Institute specializes in plant biology, while the EGF closely works together with the Centre for Mammalian Synthetic Biology at the University of Edinburgh. The UK foundries are establishing a tight collaborative environment. Furthermore, the EGF has international partners to stay at the forefront of technological development. For example, we have worked with Autodesk on a DNA design tool, and we use software from JBay. We also have special partnerships with DNA companies like IDT, Thermo, and Twist, who provide the DNA parts that we assemble. We also worked closely with Thermo Fisher and Molecular Devices on the design and integration of our automated platform. This resulted in a highly integrated platform with cutting-edge technologies that is very modular and therefore easily reconfigured to changing demands in technology. Now, Ivan will explain to you how we apply DNA assembly to the robotic work cell. We will go into details into the robotic chain that we use to build and test and we will conclude with a presentation of the software developed to support the robotic operation.
Thank you very much, Heather. So my name is Ivan. I'm the application specialist here in the Edinburgh Genome Foundry. What that actually means in reality is that my job boils down to translating the science that the Foundry sets out to do into actual robotic operations so that the instrumentation that we have can actually do it. And this includes the molecular devices, Qpix, that I will talk about in a little bit. So what I'm going to do in my segment here is start off by describing the science and the workflow that we use to assemble DNA here at the Edinburgh Genome Foundry. And then I'm going to describe the instrumentation that we have to do it. And then I'm going to specifically highlight the steps in the workflow that feature the Qpix and show how the Qpix is particularly well suited for our high throughput, large DNA assembly processes. So I'm going to start off here. What you can see here is a workflow for assembling DNA. It's one of many, but it's one that we use quite often here at the EGF. It's essentially a golden gate workflow, or sometimes called a type 2S restriction assembly method. What this basically involves is a selection of DNA parts at the very start. That's what the cherry pick part means. Once all the DNA parts are assembled in the same physical tube, a reaction mix allows them to ligate together into the final DNA product. Now, this DNA product is too small in quantity to be useful to anyone, including our customers. So what we have to do at this point is to introduce it into bacteria, what's called the transformation step. Now, this step is the beginning of our requirement for the Qpix instrument, because it will involve plating out of liquid suspensions of bacteria onto solid agar plates. And I'll describe in a little bit later how the Qpix helps us do that in a high throughput and reproducible fashion. Then, after the cells grow on the solid agar plates, we have to be able to amplify it and extract the DNA so that we can ship it off to the customers. And again, the Qpix is used in this colony picking process, and it is very, very good at picking vast numbers of colonies in a very short time. After this, we then extract the DNA from the cells that we have cultured. This is called the mini prep. And then after this, before we ship it off to the customer, we apply quality control, or QC, by subjecting the purified DNA to an analytical digest and capillary electrophoresis. This is what is referred to on this diagram as digest and fragment analysis. And assuming that the DNA passes QC, we then happily ship it off to our waiting customers. So I'm going to tell you a little bit about the Golden Gate method, or the type 2S restriction and enzyme assembly. I'm not going to go into graphic detail, but I think it does help. So the difference between this type of assembly and traditional conventional restriction digest based assembly is that it enables you to assemble multiple DNA parts together in the one reaction, and at the same time it enforces the spatial order in which these parts are assembled, so that you get assembly in a particular order rather than a random assemblage of different parts. So the difference between this type of assembly and conventional assembly is that the enzyme that is used to cut the DNA parts out has recognition sites that are separate from the sites at which it actually cuts the DNA. So if I can show you 3 different DNA parts being joined together here, what you'll notice is that the region in which the enzyme cuts, the BSMB1 site, is highlighted here with the 4 letters N. But the recognition site for the enzyme is actually separate from that. So what that means is, if you're clever in choosing the sequences that are annotated here as N, you can dictate the overhangs that are produced at the end of the DNA parts. And these overhangs enforce the spatial order in which these parts assemble. In other words, if you look at parts 1, 2, 3 here, there is no possible way part 1 could join on to part 3 directly, because the overhangs at the end of part 1 are not complementary to the overhangs at the start of part 3. So here, like I said, we can join multiple DNA parts together simultaneously, and at the same time we can enforce the spatial order in which this occurs. So here in the Edinburgh Genome Foundry, we have a specialized toolkit to do this. So we have a huge library of parts all ready to go. And what we can do here is we can assemble up to 25 different parts together in a single reaction. And we can make sure that this occurs in a specific order. So our customers essentially tell us what the number of parts that they want assembled is and what that order is, and we essentially pick them out as you would if you were selecting filling for a sandwich, and then we piece it together for them and then ship it off. So this toolkit is very flexible because it has a huge assortment of promoters, open reading frames, tags, terminators that our customers can pick and choose to suit their scientific purposes, and they can build multiple transcription units using this toolkit. So it's actually possible using this toolkit to produce up to 3 transcription units all at the same time, and this represents essentially better value for money. So here in this segment, I'm going to tell you what instrumentation we actually need to do the workflow that I've just described. At the very beginning of the process is liquid handling. So here's where we actually select the DNA parts that our customers actually want, and we pick and mix them together into different tubes, as you'll see in the animations here. So these would represent 3 different assemblies, because we can add a portion of receiving vector to each of these so that they produce 3 different assemblies in the same plate. And for this, we have the liquid handlers that you can see in the upper right hand corner. We have a LabSite Echo, which is an acoustic dispenser, and a pair actually of TCAN Evo 200s to carry out most of these liquid handling steps. We also have a bulk dispenser called the Certus. Now, once the DNA assemblies are mixed in a tube, we add some enzyme where they do their magic and assemble the DNA together. And then the next step, as I mentioned earlier, is to get this tiny little amount of assembled DNA into bacteria so that it can be amplified. And this happens during a transformation process where the DNA goes from the reaction into the bacteria, which is E. coli. And now, this is the bit where the Qpix comes in, because we have to be able to spread the bacterial cell suspensions onto a solid agar plate. So this is an example of the agar plates that we use. So here we would use 8 different regions on this plate to maximize the number of different DNA assemblies that we can fit into the same container. And the Qpix helps us do that because it has a liquid handling portion that can produce 8 different droplets on this plate. And then it has a plating head which uses 8 different pins to spread this liquid very evenly across the surface of the agar. Now this is very important because it's essential for making sure that the cells are not overly dense or overly dilute, so that they can be picked very accurately and reproducibly later. So once this is done, we leave them overnight so that they can form separate colonies like you see in this picture here. And then this is the bit where we have to select the colonies and physically pick them off the plate and put them into a new plate where they can grow and where we can use this culture to extract their DNA later. And again, the Qpix is essential for us to be able to do this in a high throughput fashion, because if you think about it, there's a huge number of colonies that are required to be picked. We pick a huge number because we have to be able to guarantee that we pick at least one that contains the correct assembly. So the Qpix, again, uses a very specialized set of heads with metal pins. They have 96 pins per head, so that means that per round of picking, it can pick up to 96 individual colonies from these agar plates, and then put them into what you see here as essentially a plate of growth medium, so that the bacteria that they have picked can grow, so that we can extract the DNA the following morning. So this is how a cartoon representation of how the picking happens. And after the growth, we would then subject it to what we call an analytical restriction enzyme digest. Now, this is the first QC step in our workflow, where we have to make sure that the DNA that is assembled is in the correct order and contains the correct parts. So we do this by cutting the purified DNA up using a collection of restriction enzymes that are selected by our software development team. Once it gets cut up, we would basically end up with a number of different fragments of different sizes. And you can predict, based on the assembly, before you actually do this reaction, what these sizes are. So then it is a matter of determining the size. To determine the size, we use capillary electrophoresis, or what can also be called fragment analysis. So our instrument is called the Fragment Analyzer. It's made by ATI. And this picture, which looks very busy here, represents 96 individual digests. So these represent 96 different clones of different assemblies. And as you can see, it is a very complex pattern. And really, I hope you can appreciate from this slide just the amount of throughput that is required here at the Foundry. So all of the instrumentation that we use, from colony picking to capillary electrophoresis to liquid handlers, they all have to be capable of very high throughput. And in a way, this is one of the reasons why we picked the Qpix as our colony picker of choice, because there are other colony pickers out on the market which produce which do the same processes, but many of them use a single pin to do this. So it is, compared to the Qpix, which uses 96 pins, they are much slower, and it's a great way to, well, it's great for me to be able to save so much time picking and plating using the Qpix as opposed to a single pin picker. So as you can imagine, with any high throughput process, we handle everything in plates, not in tubes. So here in the EGF, we have 3 different sizes of plates, which basically increase the amount of throughput that we can have. We go from 96 to 384 to 1536 wells. And at the moment, we focus predominantly on DNA assemblies using 96 and 384 well formats. But obviously in future, for the purposes of saving on reagents, 1536 would be the best way to go. So what I'm going to do here to finish off is I'm going to describe our robotic platform. In other words, the actual hardware that we use, and how it all links together in our automated platform. So what you can see here is basically a cartoon schematic of our platform. So the basic design revolves around putting all of the instrumentation, which you see in these different white shapes, and linking them all together using a series of industrial robotic arms that travel on tracks. So the robotic arms you can actually see here at certain positions on the track, And the black lines in the middle represent the tracks along which the robot arms can travel to reach the individual instruments. So I'm going to start off by showing you the robotic arms. So these robotic arms are actually provided by Thermo Scientific. They are a repurposed Fanuc robotic arm. In a different setting, you would find these Fanuc arms spray painting your car or doing spot welds. But for us, these robotic arms are very flexible, and they enable us to have the reach that we need to be able to reach the wide range of instruments that are on our track. So we have the liquid handlers that I described earlier. So these are highlighted here. We have the TCAN Evos, which produce the pipetting capacities that we would need for nucleic acid extraction, the transformations that I mentioned earlier, and a whole range of other things. For low-volume DNA assembly, so the mixing of the parts, we use the LabSite Echo 550, which is an acoustic dispenser. And for bulk filling of reagents, we have a Geiger Certus here. Now, we also have a collection of PCR machines. These are the machines in which the ligation reactions actually occur. So we have a little collection of T-robots, which are made by Biometra, and a collection of light cyclers made by Roche. We also have, of course, our molecular devices Qpix, which is located at the very end of our platform, and the back panel of the instrument is removed to allow access from the rear for the robotic arms to deliver consumables in a fully automated way. We have automated storage. Again, everything on the platform is automation compatible. So we can store consumables and plates in various different positions inside these cytomats, and we can also grow the cells that we need. So if you remember, for the nucleic acid extraction process, we actually need to grow the cells that the colony picker, the Qpix, actually picks. So they grow overnight at 37 degrees in one of these cytomats here. We have centrifuges, which are, bizarrely enough, centrifuges. And we also have a collection of plate sealers, which basically allow us to seal the plates before growth, for example. So they allow gas permeable seals to be applied to the plates so that the cells that we grow, they don't asphyxiate overnight. So the reverse of this is a peeler, and we also have a peeler that basically removes all the peels that the sealers apply. And again, all this was designed with full automation in mind, so that all the processes that you can basically do at the bench, you can also do here on the automated platform. We have delidders, which remove the lids to the agar plates. So this is a key bit of instrumentation that allows the Qpix to function in a high throughput capacity, because we can feed huge numbers of plates, agar plates, into the Qpix in a fully automated fashion, because we can just stack them into a carousel and walk away, because all the lidding and the delidding will occur automatically as part of the automated platform. We also have cold storage. These are cryovials. These are essentially individually barcoded freezer tubes in which we store the bacterial stocks in our -80 long-term storage. And this machine, this Fluidex decapper, is part of our automated solution for moving and adding the lids. We also have a flexible input hotel, which allows us to put any kind of consumable into the system while it is in operation. And we also have a barcode labeler, which allows us to barcode and therefore track all of the consumables we have on the platform. We have a large number of storage capacity which resides in our carousels. We have 3 of these, and you can store anything that you would want to use: tip boxes, agar plates, anything that you can think of in the lab in these carousels. And on that note, I'm going to hand over to Hille again, who's going to tell you about some of the specific features of our Qpix HT select system.
Yes, Ivan has explained very well how the Qpix fits into our workflow, and I would like to make this a bit more visual for you now. So this is our Qpix, and you see the automated platform in the background there. And we work very closely with the thermal design team and the OS team for molecular devices to build the first integrated Qpix. So for this, first of all, the back panel was removed so that the Fanuc robot arm had access to the Qpix. And we also ordered a Qpix 420, which usually doesn't have a plating capability, but ours has. And it also has the plating capability on SBS plates, on Omnitrace with agar, these square ones you saw in Ivan's presentation, which usually the Qpix didn't take. They took Petri dishes or Omnitrace. But we also had the option of using it manually without the whole robotic platform and use the other plate formats. It also has fluorescent imaging, allowing us to distinguish between different fluorescent markers, and also a barcode reader, because we need to track all samples. This is what the plating head looks like that Ivan mentioned. With this, we can plate out the cells in the 8 fields. And to show you how this works, we have a short movie, which I'm going to show you now, and that explains the whole process of plating. The colonies then grow overnight in the incubators, and the next day they go back to the colony picker where they are imaged, and we get this kind of image. So the colony picker identifies individual colonies, and here in yellow you see the colonies that are isolated enough to be picked, and in red some that will not be picked because they are too close together and might not be single colonies. We then can pick them with this picking head that has, as Ivan said, 96 different pins. So 96 colonies get very quickly picked and inoculated into the deep well plates where we grow up the cultures for mini prep. Just to conclude on the robotic setup and the test of our DNA assembly, we also have several other equipment to quality control and do phenotypic assaying. For example, the sequel, which is a new sequencer from PacBio and is especially good for large fragment sequencing, and also a high-throughput fermenter from N2P Labs, the RoboLector, where we can do many fermentations in parallel to test different DNAs in different cells under different conditions, and also mass spec that we use, for example, for metabolomic assaying. But I don't want to go more into detail of this in this seminar, so I would like to hand over to Valentin now, who will talk to you about the immense software efforts.
Thank you, Hilla. So we are 3 software engineers at the Foundry, and our role in a nutshell is to automate everything we can, and in particular the decision-making processes, so that the operation team spends less time worrying about the details and can focus on the big picture and developing new protocols. So we developed different kind of software for databases and tracking, for web interfaces, report generation, and very often we just write custom scripts for some specific protocols. So in this slide you can see a few tasks that are performed by software in our process. First, at the design stage, we have web services that provide information about our genetic parts registry. So customers can know exactly what parts we have and include these parts in their assemblies. Then once we receive assembly orders, which are either sequences or list of parts to assemble, we run several checks. First, to ensure that the assemblies are not pathogenic, and then to ensure that they can be manufactured without difficulties. Then comes the assembly planning, which I will speak about in the next slides. And then you have the build and test phases, for which we have software to track sample, get data files and logs from different robots, and then verify from the data we get that everything went according to the plan and that, for instance, our assemblies have the right sequence. And my next slides will be to go into more details on software operations that are just upstream and downstream of the robotic assembly. And that is how we go from customer designs to planning the assembly, and then at the end, how do we verify the final assemblies just before shipping them back to the customer. So the first step is design and design software, and we have been collaborating with Autodesk on a new online computer-aided design tool called Genetic Constructor. One specificity of this software is that it's connected to the EGF, so you can get parts directly from the EGF parts registry, and when you're happy with your designs, you can order with us directly from this interface. Once we receive the orders, we run a few checks, and in particular, we simulate the cloning that is going to happen on the robots to verify that all parts will assemble together without complications on overhangs or restriction sites and so on. And so here, for instance, you have a simulated cloning. We went from 12 parts and we simulated what will happen when we put these 12 parts on the receptor vector. So another advantage of this simulated cloning is that you get the final expected sequence of the assembly. So once you have the sequence for each assembly we are going to make, another thing we can do is choose the enzyme or the combination of enzyme that we will use to digest and to verify our assemblies. So to keep protocols simple, we try to find just one digestion that will produce nice bend patterns for all the assemblies in the assembly batch. And so that means we want patterns with at least 2 or 3 bends, and preferably bends that are in the good sensitivity zone of the migration gel. So we have algorithms that go look into every possible combination of enzymes and choose an optimal one. Another thing we can do in advance is predict how many clones should be picked for each assembly. So the idea here is that if you have a complex assembly, then you have more chances to have sequence artifacts in the end. So you should pick more clones to make sure that at least one of these clones will have the correct assembly. So the data I'm showing here is a bit old, and it was actually one of our first assembly batches. But the idea is really that progressively refine our models with the new data that we acquire from our setup. But so in these plots here, what you can see on the left is the assembly successes in green or failures in red, plotted as a function of the complexity of the assembly. So you have the assembly size, number of base pairs on one axis, and the number of parts in the other axis. And so here the correlation between the complexity of the assemblies and the success is not very strong, but globally you can notice that assemblies with less than 15 parts tend to success a lot, and assemblies with more than 15 parts have a lower success probability. So from this data, we can draw a map of expected success and determine for future constructs how much clones should get picked. And that's what you have on the right. It's a map that enables us to predict from the complexity of our assemblies how many clones should we pick. Then the last step before the actual assembly on the robots is to produce protocol files that can be read by robots. The first step to this is first locate the position of each genetic part, and for this we use an open source sample manager called ICE, which is developed by JBay in San Francisco. Once we know exactly what fridge and plate and what location on the plate each genetic part is, we need to list all the liquid dispensings that are necessary to make the assembly. So for instance, if you have 96 assemblies to do on a plate, and if you have 15 parts for each assembly in average, then you have around 1,500 liquid transfers to perform. So you list them, and then we export that list in machine format, either Chicken, Evo, or Echo, and we send that list to the robots. And so to make all this, we have a library developed at the EGF called Plato, which makes it easy to define pick lists, to export the pick list into different formats for different robots, to generate reports with maps of every intermediary plate. And also one very useful thing that it does is it simulates the pick list to make sure that we won't have any well overcapacity, or we won't have any problem with dead volumes in the wells, so that when we send the file to the robot, we know approximately what to expect, and we know there won't be any problem. Then you have the build, the proper build, and so during the build we have software to track everything, to deliver barcodes. And for instance, we have an Android app that enables you to scan the barcode, and the app is linked to our sample manager so that you know exactly the full information of a plate when you scan the barcode. So once the assembly is done, you get the first quality control data that you get is a fragment analysis. And so we're really lucky enough to have an automated fragment analyzer, and what that means is that we get actual data, not pictures, but actual data, which is the size of the different bands. So represented here is a verification of 3 clones from the same assembly. In blue on the left, you have the expected bands for this construct. Then you have 3 band patterns, 2 which appear correct in green, and one in the middle that is clearly wrong. And because we check lots of clones, like typically if you have 96 assembly, we will pick maybe 500 clones. So we try to make it as easy as possible to spot good and wrong clones with the green and red color schemes. But we also try to provide as many information as possible with these reports, so that if an assembly is consistently wrong and has consistently wrong bands, we can deduce which part may be faulty. And that's the figure you have like on the lower side. It basically tells you exactly where the enzyme is cut, so that if you have the wrong band, you can try to deduce where it didn't cut, and so what part was not inserted as it should have been. And so finally we have verification through Sanger sequencing. So here we could use, and we do use standard alignment method. That's what you have on the first figure. But same here, if you have 96 constructs to verify at once, we needed something a bit simpler to eyeball. And so we went with custom software that simply tells you exactly what your reads were, where the primers annealed, and what the perfect sequencing spans were, so that you can see at a glance if your full construct was covered and if it was right, and you can check it and mark it as done and sequenced. As a final word, at the Foundry, we try to release as much as software as we can on the internet, and so all this software that I talked about is or will be soon released on GitHub. And we try to package that software into small, very focused and easy to reuse libraries, so that you can use these libraries to build your own biological applications, even if your workflow or end goal is not exactly similar to ours.
Thank you, Valentin. So much for the software development, and I hope you got a good idea of what the Edinburgh Genome Foundry is like and possibly can do for you. Please contact us if you have any questions. All this, of course, wouldn't have been possible with a very good team spirit. This is our team here. You now have heard myself, Valentin, and Ivan, but there are also our 2 other software developers, Isaac and Aitor. Anais, our designer, who designed our logo and many other things around the Foundry, including this presentation and the movie. And Scott Nielsen, our very talented modern apprentice, and Sarah, our automation engineer. The Foundry wouldn't have happened if our co-founding directors, Susan and Patrick, wouldn't have had the idea of an automated Foundry. Patrick did his postdoc in Jeff Bilke's lab, and therefore was exposed to a lot of DNA assembly, and that's where the idea of an automated foundry was born. And we are very happy to have this going now. So thank you very much, and please ask your questions.
Thank you, Dr. Chakota, Dr. Yuen, and Dr. Zilkower for your informative presentations. It is time for Q&A. If you have a question you'd like to ask our presenters, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, do you somehow pre-screen your colonies before picking them, or is it a random pick?
Well, that's a good question, actually. Where the Qpix instrument that we have is set up to be able to do a pre-screen because it has a fluorescence turret that allows us to image all the colonies for fluorescence before. We don't currently do that, but that is definitely something that we will do in the future.
I want to advise that poll questions will appear on your screen, and you may select your answers, then close the poll questions by clicking on the X in the right corner. Okay. Our next question is, what success rate do you have for each assembly approach, and how many clones need to be picked to achieve any assembly idea?
Yeah, so that, again, it's a fairly variable number. So the success rate for the Golden Gate assembly method that I described in the webinar, that has a success rate that starts off at about 90% for a small number of parts, around 6. And obviously with the greater the number of parts you have, the lower the success rate. So that success rate drops to about 40-something% when you have a highly complex assembly, which is about 12, 20 parts. And the number of clones that we would have to pick is therefore inversely proportional to the success rate. So we would typically pick anywhere between 6 to 12 colonies per construct to be fairly confident that we would have at least one good one.
What ligase and buffers do you typically work with?
Oh, those are fairly bog standard. Ligase reaction for the Golden Gate that we described uses standard T4 ligase buffer. I believe we get that from NEB, and again, it's a very bog standard T4 ligase. It's not even a quick ligase; it's just the regular cheap stuff.
You said that the software can decide how many colonies to pick, but what is the typical number?
It's kind of like what I described earlier. It varies based on the complexity of the assembly, but typically it varies between 6 to 12.
Do you think this is an area of research that will receive more funding over the next few years?
Yes, definitely. I mean, synthetic biology is one of the really big growing research fields, and the foundry and other foundries are an essential tool for this, to develop new medicines, biofuels, and so on. So I think, yes, there will be more funding.
Where do most of your customers come from, and are you able to provide services outside the UK, say in Belgium?
At the moment, we're still a little bit in the setting up phase, so mostly we interact with researchers at the University of Edinburgh, but in the long term, we definitely expect to have customers from all over the UK and hopefully all over the world.
Does ICE generate barcodes for the plates and samples?
Sorry, could you repeat that?
Yes. Does ICE generate barcodes for the plates and samples?
Absolutely, yes. So basically the automation system, the scheduler queries ICE. So we've interfaced ICE together with the schedulers so that at runtime we query ICE, and ICE generates the barcode, sends it to the scheduler sends it to the barcode printer, and we print at runtime.
Do you do your own agar plates?
We do indeed. It can be a fairly arduous process, but we do indeed. It's a very bog standard process. We take molten agar and we pour it in a omni tray, which are the the rectangular SBS-sized containers that you saw earlier. It does have to be fairly consistent. It has to be fairly flat because the Qpix doesn't like plates, obviously, which are very bumpy or very uneven. But it is a manual process, yes.
Which software and equipment generate the information and label about the sample? And is it the ICE repository or anything else?
It depends on what you mean by information label. But if you mean what parts have to go into an assembly, yes, well, that is done by querying the ICE database. So the ICE database essentially has all of our parts. It doesn't contain information about which parts can fit together to make a viable assembly. We have the software development team has their own custom software to do that. But it all, at the software level, it all depends on ICE because the ICE repository has all of the parts. So all of the software that's written by the software development team queries ICE to get barcoding information, batch information, as well as sequencing information.
Okay, well, while we're waiting for additional questions to come in, I would once again like to thank Dr. Tekota, Dr. Yuen, and Dr. Zulkor for their presentations. Let me just see one moment. Okay, yeah, I think that will be it for questions right now. I would also like to thank our sponsor, Molecular Devices, for making today's educational webcast possible. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. And before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through October 6, 2017. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.